This recording is intended for newly assigned United Nations personnel, civilian researchers, and auxiliary security staff. If you are viewing this material, you have already been cleared for exposure to information classified under Global Biohazard Protocol. The pathogen officially designated CF01, commonly referred to as the virus, was first documented in 1922 during a geological survey conducted near a remote Siberian excavation site. Initial reports described abnormal biological samples recovered from beneath permafrost layers inconsistent with known organic life. These findings were buried, both literally and administratively. Over the following decades, fragmented Nazi research archives recovered after World War II revealed that the pathogen had been rediscovered and partially studied during the 1940s. The results were deemed uncontrollable. All surviving documentation was lost or deliberately destroyed. The virus resurfaced in 1984 near the outskirts of Moscow, following unauthorized civilian experimentation conducted by an unidentified miner, later known to the UN as containment efforts failed. By the late 1980s, localized outbreaks had spread across Eastern Europe. Public awareness was suppressed. Governments collapsed quietly. Entire population centers vanished without explanation. CF01 does not behave like a conventional virus. Infected individuals do not simply mutate, they become something else. Human biology is rewritten. Blood cells blacken. Skeletal proportions distort. Neural pathways restructure. Identity fractures. Once transformation is complete, the subject is no longer classified as human. At present, 4 primary infected classifications have been confirmed. The most common infected form, highly adaptive, social in limited groups, retains basic hunting logic. A silent stalker variant, exhibits prolonged pre-attack observation behavior, often exceeding 30 days. Aggressive, resilient, and territorial. Displays coordinated movement patterns and extreme hostility. Rare, highly anomalous, capable of consuming any known substance, retains fragments of prior human memory. The following footage documents Experiment Group Omega-7. 100 human subjects were deliberately exposed to CF01 under controlled conditions. The objective is to observe interspecies behavior when all 4 infected types emerge within the same closed environment. Specifically, this experiment seeks to answer the following. Do infected species cooperate or compete? Is hierarchy established? Can collective intelligence emerge? Does proximity accelerate mutation or evolution? The United Nations reminds all viewers, this footage is not intended to inspire hope. There is no confirmed cure. Containment remains the primary objective. Empathy toward infected entities is discouraged. What you are about to see cannot be undone. Proceed.
This is UN audio log for Experiment-1102. Experiment-1102 was conducted to observe infection divergence within a controlled human population exposed simultaneously to the A total of 100 human test subjects were selected. Subjects were healthy and infected and isolated from all external variables. Exposure method was identical across all individuals. Initial infection occurred within 9 minutes. After 72 hours, subjects began diverging into distinct infected classifications. Final results are as follows. 62 subjects developed into poltergeists. These individuals exhibited rapid neurological degradation, aggressive behavioral patterns, and the characteristic external appearance consistent with prior poltergeist documentation. 17 subjects became phantoms. These cases showed extended incubation periods, heightened predatory instincts, and delayed physical transformation. Notably, these subjects displayed prolonged observation behavior prior to full hostility. 14 subjects transformed into revenants. And like other classifications, these individuals retained limited coordination and structural stability during early stages. Social response patterns were disturbingly familiar. 7 subjects evolved into banshees. These transformations were the slowest and most unstable. All 7 cases resulted in immediate containment failure within their cells. 2 researchers were lost during the first emergence. No cross-classification mutations were observed. Each subject stabilized into a 1 infected form only.